Hello students, today we'll be continuing with the concept transgenic animals. We have seen in the previous class what are transgenic animals. Transgenic animals are those whose genome is manipulated or altered to possess the or express foreign genes, which are known as transgenic animals. They are able to express the foreign genes and they are produced by inserting the extra functional gene in the embryo or gene introduced can knock out the other specific genes that are reproducing the particular biological functions, right? And then we have seen what are the advantages of transgenic animals. In that we have seen two advantages. One is normal physiology and development. These are used to study the normal functioning of the gene, how the genes are regulated and how each gene is affecting the biological function. If the factors that are affecting the expression of these genes, how they are going to alter the particular biological process. Okay, that is the physiology of the process, how it is affected. These are all studied using using transgenic animals, that is to study normal physiology and development. And we have seen one particular example, that is complex factors such as insulin like growth factors. These are helpful in the cell growth, differentiation and fetal growth in the early postnatal stages. We have seen another advantage of this transgenic animals, that is study of diseases. That is, they are helpful in the study of diseases. They are especially mice, which has been extensively used as a model for various human diseases, and they are used to study pathological mechanism behind the disease. Okay, what are the genes that are responsible for the development of disease, and based on these studies new treatments for the disease are discovered. If the root cause is known, like the gene which is responsible for causing the disease, then we are going to see what is the product the gene is coding for and how this product is affecting a particular biological process. Based on this, the drug or the new treatment can be discovered based on the study of diseases. Another advantage of producing transgenic animals is production of biological products. It is used for production of biological products. What are these biological products? Right? Biological products are nothing but these are the products that are derived from living organisms, or the organisms itself can be used as biological products, okay, which are going to be used to treat or prevent the diseases. Getting? The biological products are derived from the living organisms such as plant cell or animal cell, and they are usually used to treat or prevent diseases, as well as they are used in other application fields also. And these biological products are very much complex to chemically synthesize them. They are naturally produced in a living system, okay, without any extra burden. But if the same biological product has to be produced externally in the laboratory, okay, or to produce in large scale, it is very difficult. It is very complex to chemically synthesize them. So therefore we are going to develop these products using genetic engineering technique. Okay, these products, they are very complex in nature, and when you are doing a large scale production also, the whatever the yield obtained is very low. So they are known as high value low volume products. They are high value, okay, they have a good value. The worth of these products is very high, but whatever the end product we are obtaining, it is low volume and it is very expensive for production if you are going it for chemical synthesis. But if you are able to express the genes coding for these products in the transgenic animals, then the cost of production is going to be less expense. Okay, the cost of production is going to be very less when compared with to produce them chemically. Okay, for example, insulin hormone, right? Now how the insulin is produced in the worldwide, it is produced from the bacteria E. coli, right? It is not, the chemical formula of the insulin is not noted down, and accordingly the chemicals are not mixed together to form the insulin, because maintaining the conditions and the formation of this insulin is very difficult chemically when compared with natural production of this insulin in the living system. So therefore the genes coding for insulin are inserted in the E. coli cells, and they are allowed to grow and express this insulin hormone. So therefore it is very expensive to chemically synthesize, but through genetic engineering technique, large scale of these biological products can be produced, and they are high value low volume products. Why? Some of the examples is vaccines, antitoxins, antivirals, okay, enzymes, hormones. These are some of the biological products. If you see the cost of the enzyme, or if you see the cost of these hormones and vaccines, they are very costly. You must be aware about the cost of the vaccine, right, which we give for the newborn babies and in the kids and all infants. When we give these vaccines, different prices are there. They are ranging from 1400, 1000 rupees, 2000, 3000, 4000. Okay, and you know how much amount of that you are going to be injected? Not even 1 ml also, it could be 0.5 ml, or sometimes it could be in terms of microliters, not even in the terms of milliliters, it is going to be in terms of microliters. Okay, means the little amount of this is also going to bring a drastic change in the body. In the body, these are not produced in the large scale. The amount of hormone, if you see your serum analysis report, okay, sugar level report, or any after analysis, if you see the antibodies, neutrophils, how it will be measured? Mg per liter. It will be measured as mg per liter or nanogram per liter. Means it is the concentration of these is going to be at very trace amounts in the body, but their effects is going to show a greater effect. So therefore the production of biological products is very important for treating the disease. Some of these are used in the medicine for treating a particular disease. So therefore transgenic animals are created by introducing a gene that codes for a specific biological product. So let us see some examples of some of the biological products that are produced through transgenic animals. So one such product is alpha-1 antitrypsin. One such biological product is alpha-1 antitrypsin. What is this alpha-1 antitrypsin? It is a biological product, okay, or it is known as a protease inhibitor. Alpha-1 antitrypsin is a protein which is acting as, the proteins can act as hormones, the proteins can act as enzyme. So this alpha-1 antitrypsin protein is acting as a enzyme, that is protease inhibitor. It is acting as a protease inhibitor, and it is used to treat the condition or a disorder known as emphysema. It is used to treat a condition or disorder known as emphysema. Emphysema is a pulmonary disease. It is caused by damage of lung tissues, thinning of these lung tissues, and destruction of the alveoli or air sacs, which makes the person difficult to breathe, and chronic cough. So it is one of the respiratory disorder, okay, or it could be due to genetic mutation also. So it is a genetic disorder. But how it is caused? When there is a deficiency of alpha-1 antitrypsin, then this emphysema is developed. Now what is the function of this alpha-1 antitrypsin? As I said, it is a protease inhibitor, and it is produced in the liver cells as well as in the bone marrow cells. It is produced in the liver cells as well as in the bone marrow cells. But this alpha 1 antitrypsin, which is produced in the liver cells, is transported to the lungs. It is transported to the lungs and it has proteolytic activity. Means it is known as protease inhibitor. So protease is the one enzyme that is going to cleave the peptide bonds. It is going to cleave the peptide bond. So it has proteolytic activity. So it is produced in the liver cells and it is transported to the lung cells, as well as it is produced in some of the bone marrow cells also. And what is the function of this alpha 1 antitrypsin? It controls an enzyme known as elastase. It is going to, because the walls of the lungs are made up of certain compound known as elastin. So this enzyme AAT, that is alpha 1 antitrypsin, controls the enzyme elastase. Elastase is the enzyme responsible for degrading of elastin of lungs. That is the reason the damage of lung tissues is going to take place, as well as the thinning of the wall of the lungs is going to take place, and therefore there is no proper exchange of gases will take place, and there is a destruction of the alveoli or air sacs that makes a person difficult to breathe in and breathe out. Right? Alpha 1 antitrypsin controls the enzyme elastase. Now from where this enzyme elastase is coming? This elastase enzyme is produced by inflamed cells. It is produced by inflamed cells such as WBCs. Whenever there is a attack of the foreign body, the immune cells are going to respond to it and produce this enzyme elastase. It is going to produce the enzyme elastase. This elastase will protect our normal tissues, the normal host tissues, from being damaged by the neutrophils and WBCs. Getting? As a part of defense mechanism, the WBCs will act against this foreign body by producing the inflammation and immune response. Along with that, this enzyme elastase is also produced. This is going to prevent the inflamed cells from being attacking our normal host cell. So this elastase protects the host cell by cleaving the inflammatory agents that are used for destroying the foreign cells. Right? So mutation in the gene coding for alpha 1 antitrypsin is going to result in the emphysema or the respiratory disorder. Right, students? Now we know what is alpha 1 antitrypsin. What is the purpose of this alpha 1 antitrypsin? It is going to treat the emphysema disease, which is a genetic disorder, and how it is going to control an enzyme elastase. It is going to regulate an enzyme elastase, and this elastase will directly, if this enzyme is not regulated, it will act upon the walls of the lungs and degrade the lung walls, leading to the thinning of these walls and later destruction of the lungs. And this elastase is produced how? It is produced by the inflamed cells. This is going to protect the host cells from immune response. Right? So this is how the alpha 1 antitrypsin is going to be function. So therefore, if there is a mutation in the gene coding for alpha 1 antitrypsin, then the body will be under the deficiency of alpha 1 antitrypsin. So therefore, using genetic engineering technique, the gene coding for alpha 1 antitrypsin is introduced into the transgenic animal, and here the transgenic animal used is transgenic sheep. Here the transgenic animal used is transgenic sheep, and the name of the transgenic sheep is Tracy. It is known as Tracy. From this transgenic sheep, the gene coding for alpha 1 antitrypsin is expressed and alpha 1 antitrypsin is produced in the mammary glands, which is extracted from the sheep through during lactation period. Getting? So the gene responsible for alpha 1 antitrypsin in human beings is SERPINA1. That is SERPINA1 is the gene that is coding for alpha 1 antitrypsin, and this is isolated from the parent cells, okay, and then it is introduced in the transgenic sheep. Okay, so the production of transgenic sheep is done by the insertion of the complementary DNA. cDNA is nothing. What is cDNA? DNA will replicate and it is converted to mRNA through transcription. When mRNA is converted to DNA, that is known as reverse transcription, and the DNA whatever is obtained is known as cDNA or complementary DNA. So this DNA coding for alpha 1 human alpha 1 antitrypsin is inserted in beta lactoglobulin gene promoter. It is inserted in the beta lactoglobulin gene promoter. Why it is inserted in the beta lactoglobulin gene promoter? So what is the function of the promoter? Promoter helps in the expression of the gene to form the product in the given specific location only, right? For example, the golden rice. So the genes coding for this beta carotene are inserted in a promoter, okay, that is associated with the protoplast, wherein the product is going to be expressed in the edible area, that is in the endosperm, right? So it is very important where you are placing the gene. So here the gene is inserted in beta lactoglobulin, which is restricting the gene expression towards the mammary tissues only or mammary glands only. So this gene will be expressed in the mammary glands and it will not be expressed in anywhere else. That is, if you know where the gene will be expressed, accordingly the extraction of the product can be done, right? So the production of transgenic sheep is going to take place by inserting the cDNA or the complementary DNA that is coding for human alpha 1 antitrypsin, which is inserted in the beta lactoglobulin gene promoter, right? Okay, then later during the lactation period, along with the milk of the sheep, with the other proteins of the milk, even the gene alpha 1 antitrypsin is going to be coded by the cDNA, and the alpha 1 antitrypsin is obtained along with the beta lactoglobulin, which is expressed in the milk of the sheep and can be extracted during the lactation period. It can be extracted during the lactation period, and the sheep used here is, its name is Tracy. So later when the yield is checked out or when the yield is calculated, the transgenic sheep is able to produce 35 grams of alpha 1 antitrypsin per liter of milk. Each liter of milk contains 35 grams of alpha 1 antitrypsin, right? So this is one of the product that is produced using biological products, that is produced using transgenic animals. Especially the animal used here is transgenic sheep. Let us see another example of, okay, first let us see how this genetic engineering helped in producing transgenic sheep, right? So here the fertilized sheep ovum is extracted. First here the recombinant DNA is produced. Getting? The plasmid is taken. Okay, in the plasmid the gene of interest is inserted. Here the gene of interest is the cDNA, that is coding for human alpha-1 antitrypsin, is inserted in the plasmid. So the plasmids used for animal cells are retroviruses. Okay, that could be the vector, or in some cases we can use the bacterial plasmids also. So in the plasmid, the gene of interest is inserted, and this gene of interest, okay, for which the vector used here contains the gene of interest as well as the promoter gene, that is beta lactoglobulin is present. Okay, now this is a recombinant DNA because it contains the vector DNA plus the gene of interest, which is a recombinant DNA. Now this recombinant DNA is inserted in the fertilized sheep ovum, which is just fertilized ovum. In that, this is first the ovum is extracted from the sheep here, and in this we are going to insert the gene of interest along with the vector DNA, that is forming the recombinant DNA. So here the recombinant DNA is introduced into the ovum, that is fertilized sheep ovum, and this DNA is injected into pronucleus using micropipette. So I have told one of the method of producing a transgenic animal is DNA micro injection. So using the micropipettes, we are going to introduce this recombinant DNA into the nucleus of the sheep ovum. Now this is implanted into the foster mother, which is nothing but the surrogate mother. Okay, we are inserting this into the surrogate mother, and then here it is allowed to take up this DNA, and it is going to produce the offspring. Okay, once the cells get differentiated, okay, now because it is transferred when it is in the embryonic stage only, so whatever the offspring produced will possess the gene of interest in all the cells, and it is going to be expressed in the mammary cells only because of the it is placed beside the beta lactoglobulin promoter gene, right? So we are having a offspring is produced, and this offspring is going to be grown enough and mature, and expression of gene of interest is restricted to mammary tissue only. So during the lactation of this new offspring takes place, okay, the milk of this transgenic sheep contains the gene of interest. I mean, the gene of interest codes for the product alpha-1 antitrypsin. So the production of gene of interest is secreted into milk of the transgenic sheep. So this is the recombinant DNA technology, or the genetic engineer technology in developing the transgenic sheep, and from the transgenic sheep extraction of alpha-1 antitrypsin is going to take place. Right, students? Now let us see another example, that is production of alpha lactalbumin. Another one is production of alpha lactalbumin. Alpha lactalbumin is a protein, okay, that is present in the milk. Human milk contains a protein which is nothing but alpha lactalbumin, and this is responsible for lactose synthesis in human milk, and also it contains high amount of tryptophan, which is an essential amino acid for synthesis of neurotransmitter serotonin. Getting, students? This alpha lactalbumin is a protein which contains or present in the human milk, and it is going to be responsible, or the functions of this alpha lactalbumin is in the lactose synthesis, that is going to regulate the production of milk, and also contains the amount of tryptophan, which is essential amino acid. So in the previous year you have studied what are essential amino acids and what are non-essential amino acids. Essential amino acids are those amino acids where the body cannot synthesize, but there is a high requirement of these type of amino acids by the body. and the synthesis of neurotransmitter serotonin from the tryptophan is taking place. From the using the tryptophan, the synthesis of neurotransmitter serotonin is going to take place, and therefore alpha lactalbumin rich milk is very important for the newborn babies as well as the adults, okay, who are malnutrition. So this genetically engineered transgenic cow known as Rosie is able to produce the milk rich in alpha lactalbumin. It is going to produce a milk that is rich in alpha lactalbumin. So it is produced from the transgenic cow Rosie. Rosie is the first transgenic cow that is being produced using genetic engineering techniques, right? And how the what is the genetic engineering behind the formation or production of this Rosie? Human gene coding for alpha lactalbumin is isolated and is inserted in cow, okay, which produced high protein enriched milk that contains 2.4 gram per liter of alpha lactalbumin, right? And it is nutritionally balanced also, and therefore it is more nutritional content is more in the milk that is produced from the transgenic cow Rosie when compared with the normal bovine milk. When compared with the normal cow milk, the transgenic cow produced this nutritionally enriched milk that containing the alpha lactalbumin, which is required for the malnutrition babies as well as elderly people or the adults whose requirement for the special nutrition is suggested, right? So these are the two examples of biological products. One is production of alpha 1 antitrypsin, another one is production of lactalbumin. Alpha 1 antitrypsin is used to treat the disease, whereas alpha lactalbumin is used to produce the protein rich milk. And attempts are also being made to find out the biological products for the treatment of diseases such as cystic fibrosis and phenylketonuria. So these are the some of the examples for biological products. Let us see another advantage of transgenic animals is vaccine safety. Another advantage is vaccine safety. So what is this vaccine safety? In order to study the safety of vaccine or in order to check the safety of vaccine, the transgenic animals are designed. The transgenic animals are specifically designed to study the vaccine safety. What are vaccines? Vaccines are the weakened live organisms. They are the live organisms, but they are weakened, and when they are inserted in the human body, it is going to produce the immune response. It will be considered as a foreign body and specific antibodies will be produced in the body, but this vaccine will, this live organism, weakened live organism will not have the capacity to produce the disease, but it will be able to produce the immune response, right? So after the production of the vaccines, they have to be tested before they are released into the market. Getting? They have to be tested. Why? Now the coronavirus research is going on, the vaccine for coronavirus, and they are still the news is highlighted as they are finding approved for clinical trials. They want approval from the clinical trials. What are clinical trials? Testing of this vaccine on human beings and studying their effects. Yes or no? But initially the vaccines are first tested into the mice and then it is tested into monkeys. If it is safe in the mice and monkeys without any allergic reactions, okay, then only it is tested in the clinical trials in a group of people. Initially a small volunteered group of people, they are going to be tested with a vaccine, and then with a moderate group of people, and then to a big population group of people. Once the quality and the safety of the vaccine is assured among these trials, then only it is released into the market, right? So since the vaccines are made with the weakened live organisms, we are not going to take any risk by directly introducing or injecting into the human beings, because human life is very very precious. That is the reason first these vaccines are tested in the transgenic animals, especially transgenic mice is used. Almost 95% of the animals are the transgenic animals contain transgenic mice. And the first for polio vaccine also, it was tested in the transgenic mice only. Why? Because it helps to check the pathogenicity. Okay, after you introduce the vaccine in the body, will the vaccine be able to produce the disease or no? That is to check the pathogenicity, as well as to know the mode of administration. Okay, if you are administering in a particular location, how it is going to be effective towards the disease. That is known as mode of administration. And studying the allergic reactions, what are the side effects produced by inserting this vaccine into the human body. All this can be studied by initially in testing the vaccine in the transgenic animals. So specifically the transgenic animals or transgenic models are designed for the study of vaccines, right? Another example is chemical safety testing. Another example is chemical safety testing. The advantage of the transgenic animal is chemical safety testing. What do you mean by chemical safety testing? Like how the transgenic animal responds to a vaccine. Similarly, the chemical toxicity level can also be studied for a particular chemical in the transgenic animals. So the transgenic animals are initially created in such a way that they are made sensitive to certain chemicals or drugs. Okay, they are created in such a way that the response of these transgenic animals towards the chemicals is going to be very quick. It is going to be very quick. So that case is created in these transgenic animals to study the toxicity or side effects of the drugs. To study the toxicity or side effects of the drugs, and whenever the gene coding for the sensitivity for these type of chemicals in the transgenic animals is inserted, since the size of the animal is small, the results can be observed very fast. The response towards these toxic chemicals or drugs can be studied very fast. So these are the some of the advantages of the transgenic animals. One is to study normal physiology and development, and another one is to study of diseases as well as production of biological products, and vaccine safety testing, as well as chemical safety testing. These are the some of the advantages of advantages of transgenic animals produced using genetic engineering techniques.